Hello, very good morning and warm welcome to all participants for second version of Global Virtual Healthcare Summit 2021 conducted by AKT Health Analytics in partnering with TAM. Thank you everyone for taking time out and coming to listen to our esteemed panelists. I am Vishal Varudkar, Director for Marketing and Sales, AKT Health Analytics, today's host for Global Virtual Healthcare Summit. AKT Health Analytics is a contract research, outsourcing and healthcare and life science strategy consulting firm. AKT Health partner with biopharmaceuticals, medical device, healthcare systems to develop market-leading strategies for long-term top-performing growth and sustainability. AKT Health enables digital transformation by delivering seamless customers experience complying to industry's security standards. We are also develop custom healthcare IT, mobile and web applications. Our objective is to provide customized quality support services by understanding your client needs to provide expert solution based on unique requirements to the clients to get product faster to the market with accuracy timelines. I welcome our first speaker, Mr. Kalwa Bhadraya, founder and director of Swaroop Tech Services Private Limited, Hyderabad. He is in chemical engineering with a specialized in the area of total quality management, ISO 9000 and 14000, industrial engineering with OR and statistics. Mr. Bhadraya has wide experience in various fields of multiple disciplinary approach. He has worked as a visiting professor at BITS Pilani and visiting faculty at Management Development Institute, Goregaon. He worked as Director for Board of Rajasthan Drug and Pharmaceutical, General Manager for Indian Drug and Pharmaceutical, and Senior Manager Technical Service at Corporate Office IDPL. After retirement in 1996, he started the mission on building a quality into the process at the level of research and development functions for pharmaceutical companies. To achieve this objective, he designed a software called SigmaTech to enable scientists to implement QBD, quality by design, with the help of SigmaTech. Mr. Bhadraya, I request you to start your, by sharing the screen, start your session. Thanks, sir. Yeah, Mr. Bhadraya, you can start the session, sir.
Yeah. Is it visible now? Hello?
Visible, sir.
It is visible now?
Yeah, you are visible. You can start, sir.
Why QBD is a must for pharma industry?
Can you play it, sir?
Yeah. In pharma industry, the obsolescence of medicine is very fast, relatively. With the result, the companies always work on finding new products and process to compete in the market. This is why R&D has become a must in pharma industry. But unfortunately, many pharma industries follow good old methods of process development by resorting to traditional methods like subjective methods, hit and miss method, trial and error method, O-factor, that is one factor at a time method. These methods will not enable the scientist to achieve the least cost, best quality, and least delivery time, which are the cornerstones of the business organization to compete in the market. The pharma companies incur a cost of non-conforming to the quality to the tune of 25 to 30% of sales turnover as a result of these traditional methods. Since these costs are hidden across various departments, they cannot be seen so easily, and the survey shows that the cost of not conforming to quality are huge. Best observed one in the traditional method was taken as a optimal, but in practice it was not possible to attain consistent results due to the variation that violated the specifications. Tighter specifications for raw materials and operating parameters were imposed to obtain a better result. Even the better results obtained were with increasing costs. In fact, the factors affecting the variation were unpredictable basically due to OFAT and other conventional methods. When the quality preference came on the scene, then it was intended to build the quality into the process. Then all sort of in-process inspection, statistical control methods, etc. started. Okay, but then what happened? But throughout these stages, focus was more on the inspection only through the different methods. Again, it was forgotten to build the quality into the process. So rejections at incoming materials, in process level, and final product level reduced, but could not be to the desired level of reduction in defects. The quest for quality continued. The quest to quality question whether the given process is capable of giving a defect-free production. Then came another methodology called process measurement and process capability index came into the limelight. On measuring the process capability, we could only know at what level the existing process is running and the level of consistency in the process. But still, the question remained how to improve the process capability for defect-free production. With the application of SQC and process capability, only the identification of assignable causes and inherent causes could be established. But how to remedy the assignable causes became a problem, particularly at production process. See, in the various methodologies like philosophies started getting implemented like benchmarking, BPR, QC, TQM, TPM, etc., ISO, to eliminate the defects at the source level like men, materials, methods, and machines. Though the improvement has been there substantially at the operational level, at production process level, not much dent was observed because of technological issues. Motorola was trying to improve the quality by various methods, but could not reach the Six Sigma level quality. And when they applied DOE statistical design of experimental methods, they could quickly achieve the Six Sigma level, a highest level of quality. Then DOE came into limelight about the benefits of DOE. DOE means design of experimental methods, statistical designs. Because of two, okay. Yeah, in fact, when the data was submitted by the pharma companies to the regulators, they were asking the scientists what happens if we go beyond or below the set points submitted. The scientists were not able to answer to the satisfaction of the regulators. The regulators felt that the dynamics of the process development is not understood, and hence they felt a new approach required for the complete understanding of the process dynamics to develop a robust process with a wide range of parameters where the quality will not suffer. Keeping this in view, ICH, to which USA, Europe, and Japan are members, have formed certain guidelines around 2000 for the process development to mitigate all these difficulties. By adapting to QBD, international market acceptability will be forthcoming and easy to move exports around the countries like USA, Europe, Japan, where our maximum efforts are taking place from India. ICH guidelines for QBD have been adopted as law in several countries, but are only used as a guidelines for the US FDA. The same have been issued by FDA in 2006 production by the pharmaceutical. The tools of the QBD for the implementation of ICH are ICH Q8, dealing DOE application, design and development of the robust process. ICH Q9, implemented quality risk analysis. ICH Q10, implemented quality systems and CHMP. ICH deals with the DOE as a tool which requires software for implementation, and it is primary tool scientists to use it in designing and development of a robust process. Robust design methods refer collectively to the different methods of selecting optimal targets for the inputs. Generally, when one thinks reduction in variation, tightening tolerance come to the mind, which is costly affair. But as demonstrated by Dr. Taguchi, variation can also be reduced by careful selection of the targets. Some of the methods of robust design are dual response approach, Taguchi method with S/N ratio, simulation of process models and graphic interface, design space, which is required by for the regulators when you are submitting the Six Sigma approach, measurement of process by process capability index, and applying DOI technique to improve the process. So today I am going to show the demonstrate one application of this measurement of the process by process capability index and applying the DOI to improve the process. This is about theoretical background of the QBD. Now we'll go for the application part of it as a case study. Is it visible, Mr.? Hello? Hello? Is it visible?
Yeah, it's visible, sir.
Okay. This is introduction about this process measurement and process improvement. A case study with the application of these techniques, process measurement and improvement in process industry is very useful to the process engineers and production engineers to understand the concept. How to define, measure the process, analyze and improve and control, it's called DMAT, in process industry is shared with a case study presentation. Define. One of the penultimate stage in a drug manufacturing process called pyramidal, the yield was too low, around 40% against an expected yield of 60%. Since in the process the isomers are formed, the desired isomer cannot be more than 60%. There is a huge gap between 40% achieved and 60% expected. The process chemists have been telling that all the operating parameters like temperature, pH, time, and etc. have been standardized, and as per the SOP, standard operating system, they have been producing it. Even then, the variation was too high. This became a bottleneck for the capacity. It was all the more essential to improve the output measure. To demand the process measurement of the existing process, an exercise was performed by collecting the data of output from the batch sheets of the process and was measured. Okay. See, the process capability of the existing process. The average was 40.528 and upper specification was achieved only 43 and standard deviation 1.8060. Now the CPK, process capability index, is as per this formula, it is just about 0.457, which is near 2 sigma level, and corresponding COPQ, cost of poor quality, is 25 to 30%. This is a too costly affair because this is very too quality level. The quality inbuilt was not at all sufficient, That's why mostly, since CPK is less than 1.33, it infers that it is not possible to achieve the targeted yield output in the existing process. Hence, it requires to improve the process. Identification of the input variables for the process. On proper analysis, the input materials ratios were formed, grouped into 3 mole ratios. One is M1, mole ratio of beta ethoxypropanediol to sodium. Then M2, number of moles of ethyl formate, and M3, acetamidine hydrochloride to DMS, as a mole ratios. And whereas sodium was kept as 1 mole constant. It is required to conduct a fresh study to conduct the experiment to find out the factors affecting the output. The factors and their levels are given in the table below. Then DOE. Since already OFAT was ruled out for obtaining satisfactory results, DOE method was suggested for investigation by experimentation. This is the most powerful method for investigation of factorial effects. See, this is the Sigma Tech software. You can see, click on problem selection. Then you will find optimization model, screening model. Then ours is a 3 variable model in optimization. So I click on the 3 variables. Then I already stored the data in it. So I am opening this application. Then this is the name of the project and output is written there. Here, name of project and output, it is expressed as a yield in the process industries. Okay? Then save it and click on next. Then this is the variable entry table. This is M1 mole ratio, M2 mole ratio, moles, and M3 mole ratio. Units, numbers, this is low level, this is high level. Okay? Then click on a plan. See, this left side plan is given by the software. Once you enter that input variables, then see what is happening here. 8 experimental permutation combinations have been given by the software. In addition to that, 4 replicate experiments are supposed to be done at the midpoint of these variable levels. And all the parameters also have been given by the software. Accordingly, the experiments were conducted. Their output as yield is recorded in the output column here. Now you will see here, when X1 and X2 were increased here, there was not substantial increase. But when X1 and X2 simultaneously increased to higher level, the output jumped to 56. Okay? This is called positive interaction or synergetic effect of the interactions, which is helping to develop the process, improve the output. Then go for the analysis. This is called statistical analysis. What is happening? We wanted to develop a model y is equal to b0 plus b1 into x1 plus b2 x2 plus b1 in x1 x2, etc. Then for that we need to evaluate the coefficients, that is constants of this b1, b2, etc. All these things have been evaluated by the software. And there SS ratio, this is sum of the squares ratio, it is called in statistics. It is nothing but contribution to the process by each variable and their interactions. Now you can see x1 and x2 both have got positive contribution, just around 5%. But their interaction, x1 and x2 has got a coefficient is 3 times to this and contribution is 33.5%. This is a very healthy sign, okay, to improve the process. Then x3 coefficient is minus 1.8 and SS is about 49% contribution. So that means higher the mole ratio of x3, lower will be your output because it is a negatively contributing. So our strategy is to increase the x1 and x2 and reduce the x3 for optimization. And it also has calculated nonlinear effect. Whether nonlinear means the output will go as the variables inputs increase to some extent, then it will come down. It is a curvature effect is there. So the software says the nonlinear is very significant. If it is significant, we have given the guidelines here what to do. Then as per this guideline, we have to simply click on the composite here. So the composite design, these are the composite design. The moment you click on a composite design, it is asking to conduct 6 more experiments and their parameters have been given. And as per this, again, the experiments were conducted and outputs are recorded here. You will be surprised to know when we have increased x1 and x2 more higher mole ratio, we have got this 56. Now here, when all these mole ratios are less than those things, previous one, this input quantities are quite less than compared to previous one, still it will give 56. Because of the non-linear, this phenomena will be happening. So this is a very good sign. I mean, even with the lower inputs, you will get the higher outputs. Then click on the analysis. Now you have got the quadratic model completely developed here. This is earlier already we have developed up to this. Now x1 square, x2 square, x3 square also have been estimated here. Now full quadratic process model is available and no more experiments need to be conducted. Okay? And now we go for the simulation to achieve our expected results. In the simulation, what happened? It has completed about 25 simulations. Automatically it does it, and it is given output also here. Response output is given. So it has given the 60% of achieved one also, and others also are given. So to understand it, basically, I have given a graphical interface here. Now you see, this is x3 has got a negative coefficient, so it is kept as a constant at lowest level. And now we have got 5 levels, x1, right from this level to this level, 5 levels are there. And x2 also 5 levels are there. Each graph is for each one line of one level of the x2. Now you can see 1, 2, these two are coming down, so we are not interested. Now there is one thing going very steep, these and these two. That is x2 is equal to 1.96 and 2.18 are going to give the same result, x, that is 60% at x1 is equal to 1.091. But this is what happens only at a single point here. Now we wanted a robust process where even input in variation in the input should not suffer my outputs. Now you see this part of it where x2 is equal to 1.74, it goes slightly up and then remains almost constant parallel to the x-axis. That means even when you are changing input quantities of the x1 right from 1.023 to 1.091, your output is remains almost constant, 57 plus. Okay, this is called robust process parameters. That means even there are changes here in this input material, your quality will not suffer. And this is called robust process parameters, which will give you 57%. Where again the 43 which was obtained in the plant, now this is, and it has been confirmed and again by the experimentation in the lab and implemented in the plant. Okay. Now we are again confirming whether robustness is achieved or not mathematically. Okay. So by the for that we have calculated the CPK. CPK has come to 1.85 and which is equivalent to 5.55 corresponding that is sigma level. And CPK highest level is 2. And this 5.55 corresponds to about less than 10% of the cost of poor quality, as low as 5% even. So this is how we can measure the process and improve the process as expected. And you can also verify whether we have received the that is what you call the robust process or not. So all these methods are available. Okay. So any questions you would like to put it? I try to confine to the 30 minutes time, and still I am leaving some time for the questioning.
Thank you very much for a wonderful session on QBD, Dr. Bhadraya. And I request a participant, if you have any Q&A, you can post it through Zoom so that we can able to ask. Sir, I have one more query that, so how many variables is the software is going to take, sir?
Oh, it has got up to 23 variables. See, we have got two models. One is the screening models, other one is the optimization. Optimization module can work up to 5 variables presently, okay? And screening modules, it will go up to 23 variables. When you have got, say, about 10 input variables, you don't have to conduct to hundreds and thousands of experiments. You have to conduct only 12 experiments. And it will sort out as per the Pareto's law, what are the significant contributing factors to the process. And that will come out as per the Pareto's law, only 20% of it. So that comes to only 3, 4 or 5, something like that. And that 3 or 4, 5 will go for the optimization, like that. So first we will go screening and then go for optimization. if there are too many variables.
Okay.
It will like
Okay. There is one more question. So, which organization is Sigma-Tech providing software already, and what is the success rate by using the software, sir?
See, this we have been applying to, given to many leading organizations like Ranbaxy, Freshness Kabi Oncology, which is a Israel company, which is one of the highest biocontrol company, and Freshness Kabi Oncology, it is a German company. Then we started with Dr. Labs also quite in 2005. Then Aravindo. Then recent one is the NATCO and Suven Life and Newland Labs here in Hyderabad. And Micro Lab also we have then given it this what you call your Bangalore. So like that, many industries we have covered about 25 institutions and corporate R&Ds.
Okay, great.
Because unless you develop a robust process R&D, the defects are going to continue on the production line. You cannot change in the production line. You have to build the quality only at the R&D level, not at production level.
Okay.
Production level automatically it goes. Once you do it right process at the R&D level.
Okay. Great to know that, sir. And we got one more query from my end, sir.
Yeah.
Actually, when you have been doing in a software and when you have been doing reality, so is there any percentage of change when you have been doing in software and when you have been doing in really when you have been like in analytical when you have been doing? So what is there any difference or it shows exact results?
No, see what happened when we have given a simulated runs and we have split up a most optimal one, robust process, that they will again experiment and confirm for the results. Okay. After confirming, it will definitely confirm because there is a measurement of the process model also as an R square we call it, whether it is going to give the correct estimates or not. 99% and above it will give the correct estimates. It is as per that statistical prediction. So they are giving correct results actually. Otherwise, nobody, see, these people would not have come forward to go for this software, isn't it?
Okay.
And they are implementing already.
Okay.
And I have given the consultants also to the QBD project to the Newland Lab just last year, And after coming, once the COVID started, then we were not able to move further again.
On Sigma-Tech software, any publications based on the project software?
Yeah, plenty are there. See, applications you will find in various international journals are there. I can share with you about that document also.
Okay, sir.
And particularly, many research scholars also have been taking the advice from me how to use this Sigma-Tech. And we have worked with them and submitted the paper, and they have been published in the international journal. And I too have published in some of these things into the different journals.
Okay. So it has been done nationally and internationally. It got published most of the papers.
Yeah.
The help of software. Yeah. Sir, it was. Thank you very much for being a part of our Global Virtual Healthcare Summit, sir. Thanks for your time and giving us a valuable knowledge regarding Sigma-Tech software, which can able to support pharma industry to develop a robust process and come up with a solution with even a more number of variables up to 20 and giving the solutions to the industry and making the product come to the market in a quicker way, sir. Thank you very much for your time.
It is basically user-friendly. Sigma-Tech is a very user-friendly. The design is like that. As you are carrying the R&D process, it's in the same fashion it is designed.
Okay, sir. Thank you very much, sir. I request you to stop your video so that the next participant can.
Okay.
Yeah. Thank you very much, sir. I request Dr. Praveen Kumar, our next speaker for the session. I welcome you, Dr. Praveen Kumar, for second version of Global Virtual Healthcare Summit conducted by AKT Health Analytics, partnering with TAM, sir. And Dr. Praveen Kumar is an experienced associate professor and reputed bioinformatics.
Yes, good morning, Vishal sir. My video as well as PowerPoint is visible presently. Can you able to see my PowerPoint?
Sir. Yeah, I can. Wait, sir. Actually, I think you have been stopped. Sir, give a second, sir. Give me a second. Let me introduce
yes,
the participant. Then you can share your screen, sir. Yeah. Dr. Praveen Kumar, wait for 2 seconds. Yeah, Dr. Praveen Kumar is an associated professor in and bioinformatician with a demonstrated history of teaching in biotechnology field for 18 years in reputed engineering college in and around Chennai. He worked as a bioinformatician tool, including machine learning, AI, chemoinformatics tools with wet lab experience on HPLC, ELISA, FTIR, and small animal handling experience. Dr. Praveen Kumar has been published 24 papers in drugs and vaccine design topics in reputed journals like scientific reports in Nature Publishers, Mini Review, Medicinal Chemistry in Bennett Science Publishers. And he has been conducted many workshops like STTPS, FDPs, seminars, webinars, and certification courses with funding and sponsored by ICMR, CSIR. He has received a mentorship award from offices of Principal Science, Government of India, Minister of Education, and he has received a Best Research Award category for 12th and 11th International Scientific Award 2020 for a very good organization. Thanks and I request our participant, you can post your Q&A so that at the end of the session, Dr. Praveen Kumar can answer the questions. Sir, now you can share the screen, Dr. Praveen Kumar.
Hello.
Yeah,
Am I audible to you, sir?
Yeah.
You
are audible, sir. Can you share the screens?
Yes, sir. I am. Is it visible, sir? My screen?
Yeah, visible, sir.
Good morning, sir. So good morning to one and all present here for the World Summit. Thanks for providing my brief introductory note, Mr. Vishal sir from AKT Healthcare. So I'm proud to present in this World Summit 2021. So I'm going to present on the drug design for SARS-CoV-2, especially with the target receptor spike protein. So my title is on identifying covalent spike protein inhibitors to control the infection of SARS-CoV-2, which is presently ruling for past 3 years. So I've taken this topic for presentation. In humans, coronaviruses have caused several respiratory infections, starting from the cold to more severe diseases like Middle East respiratory syndrome and severe acute respiratory syndrome. The spike protein of the coronavirus, which is most important, which shares almost 57 and 82% homology with the spike protein of SARS-CoV-2. COVID-19 was declared as pandemic globally on 11th March 2020 by the World Health Organization. The spike glycoprotein plays important roles in virus attachment, fusion, and entry into the host cell. The WHO report, as on 2 days before, I just taken from the WHO website, it shows that it reveals that 235 countries have been affected by COVID-19 with over 214.5 million people affected worldwide and a fertility rate of 2.05%. However, in India, we are surviving with a fertility rate of 1.31%, up to 32.7 million people affected. And if we have a comparative study of most occurring viral diseases, COVID-19 and malaria, when compared to number of cases affected globally by COVID-19 disease in past 3 years, the number of cases affected by malaria in the past year alone is maximum. Okay, it is 229 million when compared to COVID-19 for past 3 years, it's just 214.5 million. but percentage fatality rate of COVID-19 disease is 2.05 percentage, and is more when compared to percentage fatality of malaria disease, which is just 0.17 percentage. Which these statistics have taken globally from, of course, from WHO, and up to in this slide we can see up to present 11,751 literatures are published on computational studies in COVID-19 for past 2 years. And if you see here, the 1.63 lakh literatures are published in NCBI PubMed. This is an portal everyone know, standard journal articles are published with respect to biomedical and biotechnology kind of a domain. Like PubMed and Medline works together to have these kind of a literatures. And if you see in malaria for past 200 years, okay, we just have 1.02 literatures. Then I think from this slide itself, we can realize how much efforts the scientists and globally has put to discover drugs as well as vaccines for SARS-CoV-2. And if you see here, the computational alone, it comes around up to 11,000 literatures. In the next slide, we discuss about the target receptor databases. Especially, we know PDB website, okay, that is the repository of 3D structure of proteins. Generally, these repository, we can find the proteins are deposited using NMR or X-ray crystallography studies. And if you come across basic information about SARS-CoV-2, the SARS-CoV-2 is a linear, positive sense, single standard RNA virus. And if you see the genome size, it comes around 29.9 kilobase pairs. So this is the size, genome size of SARS-CoV-2. And if you see, there are totally 20 important proteins are present in SARS-CoV-2. Out of 24 are structural proteins. The four important structural proteins are spike, envelope, membrane, nucleocapsid. If you see, the most important target receptor is the spike glycoprotein, just because it plays an important role in virus attachment, fusion, and entry into the host cell. And other than this, we also have 16 non-structural proteins, that is NSP1 to NSP16. And this is the 3D structure database called PDB. Up to present, we can see the 1.72 lakh structures of proteins, the 3D structures of proteins are deposited using the standard gold standard techniques like NMR and X-ray diffraction or X-ray crystallography. Also now presently, cryo-electron microscopic techniques come forward to publish more of 3D structures in this database. And if you see, other than the deposition of these SARS-CoV-2 14423 structures, we also have a few more structures, few more proteins related to SARS-CoV-2, which we can find somewhere here. And if you see in this slide, overall structure of SARS-CoV-2, the receptor binding domain bounding to human angiotensin convertase enzyme 2. So this, if you see in the green, the ribbon shows the human ACE2, and the blue color, whatever we find here is the spike glycoprotein, which we simply abbreviate like S. And you know, the spike glycoprotein generally try to target the host in the receptor binding domain. If you see the receptor binding domain, okay, of spike glycoprotein, we have almost 13 important binding sites, which binds with the human ACE2. So if you go for drug design, so in the target receptor, we have to focus on these binding sites. And if you see this kind of receptor binding domain, it comes around 319 to 541. But if you see more specific, which is having more hydrogen bond interaction, we can, instead of simply restricting up to RBD, we can also go for receptor binding motif. This is more specific, and it comes in between this region, that is 437 to 508. So in this 65 amino acid region, you can find more kind of a specific contacts. So not only you find more bonding, also non-bonding contacts like hydrogen bond contacts will be more in this region. So we have to target that kind of receptor binding domain. And if you see some complexity with respect to spike glycoprotein, we have two different types of states, open and closed state. And if you go for the the spike glycoprotein can be in a receptor inaccessible in a closed state. Okay, but if you see the open state, this is more accessible. Okay, the open state is more accessible when compared to the closed state, which is inaccessible. So, and if you further come across that is if you can do the transition to the semi-open state, which is of course provided as a crucial step or critical step in host cell recognition of SARS-CoV-2. Okay, here this is the semi-open state. If this is closed state at the top and this is the open state mentioned in bottom, and this is the semi-open state, just because it is in the semi-open state, the spike glycoprotein can able to interact with the receptor binding motif domain with the human acid. Or if it is in down state, you can't able to recognize it. Okay, so the recognition with respect to spike glycoprotein is a very important step. And also, while going for drug design studies, we take care of both closed and open states, and we check whether we are docking in the exactly like a semi-open state. And in this slide, we can able to see the spike mutations of SARS-CoV-2. So till now, eight important mutations, I mean, 8 different variants of SARS-CoV-2, we can find some kind of multiple point mutations. And if you see the 8 variants, okay, the first variant is alpha, second beta, third gamma, the fourth is delta variant of spike glycoprotein, eta 5, iota is 6, kappa is 7, and lambda is 8. So out of these 8 variants of SARS-CoV-2, the delta and the kappa is originated from India. So this data I have taken from CDC government website. And if you see the mutations for the 8 different variants, what I've shown, okay, for alpha variant, okay, in the spike glycoprotein, we find 4 important mutations. Okay, so 4 combination of mutations we can observe, and similarly in the beta, we can find almost 8 combinations of single point mutations with respect to spike, and gamma 3, delta 4, zeta 1, lambda, nearly 7 types of mutations are there. Okay, and in eta variant, if you see the specific region, like in 23 countries, we observe these kind of mutations with respect to eta variant of SARS-CoV-2. And for theta, we have 4 kind of mutations, and we observe in 18 countries. And the lota, we observe in 18 countries. And in kappa variant, we observe 54 countries. Now, whatever we speak, the second wave, the transition of second wave to the third wave, okay, so this we speak with respect to delta variant. Okay, nearly 4 different mutations we are encountering. And if you see the delta variant, it was observed in India, and it was reported on October 2020. And so after third wave, so like from my data, it is evident that we may come across even 4, 5, 6, already the fourth wave, okay, hit badly in Tunisia, Africa. And the third wave is badly hit in 6 different regions of Africa. Okay, but in India, it is a slight kind of hit is there, but we may experience in next month. Okay, if it is taken in proper control, we may can avoid much kind of fatality in India. Now, overall, you can able to find when compared to world, in India, it's the percentage fatality is very less, isn't it? So now, if you see the computational methods in drug design, well, I've mentioned you nearly 12 different methods. So recently, like in last year, the AST India come forward with a kind of a competitive event for students. They have conducted a drug discovery hackathon where they asked us to display these kind of methods. Okay, and whatever methods I have shown, like drug likeness prediction, ADMET, and I have shown you the freewares to interact. Okay, so even any the students can try to use this kind of a freewares other than a few software. These softwares are commercial, of course, But most of the softwares, whatever listed in the slide, are freeware. So we can try it in our system. And similarly, if you see the small molecule databases, we have NCBI PubChem. Up to present, 1,500 antiviral compounds are used globally to treat the COVID-19, where nearly 2,278 pathways are there for treating this COVID-19 disease. So the COVID-19 linked with various kind of disease, respiratory, cardiac, etc. And if you see in this next slide, the Chem Spider is of course a database of a repository of organic and synthetic chemical molecules. CHAMBL is one more database, and the URL is mentioned, where you can find the 3D structure of compounds and the properties of compounds. Also, we have CHEBI and drug bank. This is the database where we can find more drug molecules deposited. Not only we find just the 3D structures, we also find some kind of information like the annotation, like chemical identifiers, clinical trials happen with respect to drugs on pharmacoeconomic spectra, targets, enzymes, etc. For finding the binding site information and few more physicochemical properties, we can search in binding DB database. KG is one such database where we can not only we can download the 3D structures, we also find some information related to pathways, enzymes, drugs, etc. And if you go for the first method like drug likeness prediction, we can predict with freewares like MolSoft, Mol Inspiration, Swiss ADME, where we take care of molecular weight, the first property, where according to the Lipinski rule 5, also we call it as a Pfizer rule 5, it should be the molecular weight of a compound should be less than 500, and the partition coefficient should be less than 5. Hydrogen bond acceptor should be less than 10, and hydrogen bond donor should be less than 5. These four properties we majorly take care of in Lipinski rule 5. Like if any compound has to be treated like a drug, okay, it has to satisfy any 3 properties out of 4 properties, whatever I have mentioned. So other than these 3 properties, we also have few more properties in consideration to check the drug properties in the various kind of rules, like polar surface area, rotating bonds. So even these parameters we can consider to check whether a compound can act like a drug or not. To find the drug likeness, we can use MolSoft software. Kind of a output is mentioned here from the and ADMAT properties. This is the one more method we call that as an absorption, distribution, metabolism, excretion, and toxicity studies can be done using this software. Okay, for this we can use freewares like Swiss ADME and few more softwares mentioned. And one such output is shown from the Swiss ADME. The major parameters what we consider in drug likeness by Lipinski rule 5, the same properties we can observe. For example, if you see the hydrogen bond acceptors here for a compound, whatever is shown should be less than 10. It is showing here as 6, which is less than 10, and then hydrogen bond donors is showing as 10, which is less than 5. And if you see the molecular weight, okay, that should be less than 500 gram per mole. Of course, we got 363. Okay, and the last parameter is the partition coefficient. The partition coefficient should be less than 5. Of course, we got 3.2. So just because in this picture what I shown, all the 4 properties are satisfying, we can tell this compound can act like a drug. I think I have shown a drug of a luteolin compound. Okay, now I think we can go for the next slide. So just like ADMAT, we can also go for you can all like Swiss ADME, we can also use pre-ADMET to calculate the ADME, not only ADME prediction, also we can check the toxicity as well as the mutagenicity studies. And if you see in this slide, this is click to drug website where you can see the pipeline of how to discover the drugs, okay, using various methods. So this kind of a pipeline helps you to do what kind of method you can use to analyze the drug properties. And in this slide, we can able to see a kind of event which happened in 2020, and the results were declared on 16th February. Like a meeting was conducted on 16th February, and within 2 days they have declared the result. So I've guided a team of nearly 6 students, okay, 6 UG students, second year UG students, those who don't have even bioinformatics skills, but later they have learned during the COVID-19 lockdown situation. Not only they learned bioinformatics, also learned the drug design skills, and they competed almost with 12,000 teams. They have competed with 12,000 teams all over India. And this event was organized by the Office of the Principal Scientific Advisor of the Government of India, ACT India, CSRGOI, MHRD. So out of 12,000 teams, only 53 teams got selected for internship. These students are almost getting nearly 3.6 lakhs as a stipend amount winning the competition. And so when they have taken this topic to present for this World Summit, so we can come across what or what exactly is the methodology and what results we got for with this kind of a project, a small project we done in our institution itself. And if you see here, the methodology, what we have taken is we have taken almost 1,152 compounds at the last year, where we found it as an antiviral compounds deposited as a data set we have downloaded from NCBI PubChem. Okay, so as such, we have taken the entire compounds and we have filtered almost nearly 400 compounds by checking the Lipinski rule as well as ADMAT properties. Out of 400 compounds, we further gone for screening using virtual screening by Autodoc Pyrex. From Autodoc Pyrex through the scores, so by getting the negative maximum scores, okay, in terms of kilocalorie per mole, so we screen further up to 100 compounds, and we have taken 100 compounds finally to dock with Autodoc software manually. So whatever 100 compounds we have taken, we docked with two different states of spike glycoprotein. One is open and another is closed. And we also check whether is it binding exactly at the receptor binding motif or not. Okay, so this is a kind of a methodology we have used. Other than the docking, we also gone for the simulation, which they didn't require, but still for showing the stability and for betterment, we have also gone an additional step. After docking, we pass the docking complexes for the simulation. At last, we have scrutinized the three best compounds. The first best compound what we obtained is a vithaferin compound, which is binding with almost two binding sites of the receptor binding motif. Abivatinib is the second best compound. The luteolin is the third best compound. About vithaferin A, this is a compound we can derive from Ithania somnifera, also called as Indian ginseng. We call this as an Indian ginseng. We simply in Sanskrit or mostly we call it as an ashwagandham. This shows a better kind of a property, showing the better binding results and showing the better binding sites. And similarly, abivatinib can be derived from Cannabis sativa. This is also one of such Indian plant. Okay, also showed these compounds already showed drug-like properties and are used for treating SARS-CoV-1, which occurred some 15 years before. Okay, but also luteolin. This is also one more compound which obtained from an Indian plant called Hydnocarpus vegetariana. So almost all these three plant compounds we can get. Of the simulation results, in the simulation results, we can able to find our ligand is found to binding stably with the protein at 0.95 nanometer for vithaferin. And if you see for the abivatinib, it is at 1.5 nanometer. And if you see for the abivatinib, it is at 1.5 carbon fluctuations observed in RMSF, and also the energy is found to be stable in with both the plant compounds. And overall, with respect, if I would like to conclude, the spike protein plays an important role in viral entry by interacting with the human AC2 receptor. And if you see the transition to the semi-open state is a critical step in host cell recognition of SARS-CoV-2. So according to the problem statement we have chosen for drug discovery hackathon, where my students cracked, okay, they identified three important compounds with vithaferin A, abivatinib, and luteolin. Compounds are screened in virtual screening, and of course in docking and simulation we got a kind of a results where we found the compounds are binding stably. So these compounds are also cleared phase 2 trials, and these are some of the references. And about my institution, our institution was established in, my Department of Biotechnology in SVC was established in 2006, and so presently up to from up to present we are having almost
Dr. Praveen. Hello.
Hello, am I audible? Am I disconnected just now? Or
Yeah, it was a bit. Yeah, you continue, sir.
okay, sir. I don't know how much
Yeah.
whether I have completed this slide, sir. The summary slide.
Yeah, not yet. You can share the screen, sir.
Oh, sorry.
Yeah.
I think I have shared the screen. May I know up to where you have seen, sir?
The
Up to
conclusion,
this slide.
the conclusion. Summary.
You have seen this slide, sir?
Yeah,
Summary. Okay. Okay, I conclude with the summary. The spike-like protein plays an important role in viral entry by interacting with the ACE2 receptor of host cell. Transition to the semi-open state is a critical step in host cell recognition of SARS-CoV-2. So, from the problem statement chosen in drug discovery hackathon organized by Government of India, my students cracked and found out in the project that vitafenib, abivatinib, and luteolin compounds, which we can obtain from the various sources of India. Can be used as a plant-based drugs for SARS-CoV-2. And in the docking results and simulation results, out of 3 compounds, vitafenib showed a best compound for treating the SARS-CoV-2 infection. Of course, IIT Delhi has taken this kind of a plant and compound to go for the phase 3 clinical trials. And of course, almost all the 3 plant compounds also cleared the phase 2 clinical trials. And these are the references. And up to my institution, the Department of Biotechnology, SVC, was established in the year 2006. And up to present, like in the 15 years, we got nearly 1.72 crores from the various government funding agencies. Also, we have done we got like extramural research grants, international travel grants, research consultancies, grants, etc. Now it's up to the question round. So if you want to if you have any doubts, I can question.
Yes. First of all, sir, thank you, Dr. Praveen Kumar, for wonderful session on SARS-CoV-2. I request audience or attendees to pose their Q&As to Dr. Praveen Kumar so that he can able to answer you and can guide you for your future course. So it is almost related to our COVID. It's mostly even our present situation in India and globally, it is like COVID-19. So it's mostly related this Indian hubs, what you have been identified, can able to give us an immunity towards COVID, sir.
Yes, sir. Actually, sir, whether you can able to see my screen or I just close the screen. No.
Yeah.
Okay. I already shared the screen, isn't it?
Yeah.
So, okay. I just explain you, sir. Presently, we are facing the Delta variant, and in this September, just because in each and every day, okay, we are creating a new records of taking more than 1.23 crore doses in a day. So this is the country, okay, where we can find more vaccine production. And of course, just because of more vaccine production, we can able to bring down the percentage fatality from overall fertility from 2.05 to 1.31%. So from my side, I can provide you some information. That is, we are facing the Delta variant, which is originated from India, but this is slightly suppressed because of more vaccination doses happening on in a day. But only the Kerala and some northern parts, okay, only few parts, or like Nagaland, etc., are suffering with the third variant, that is the third wave Delta variant. But it will be come into control. It will be stopped. It will be declined within a few days. So almost, I think we can overcome the third wave, but we don't know what happens with the next month. Okay, so even if we experience a kind of Eta or the Lota, that kind of a variants, we have to check how much vaccine is putting the efficacy for these kind of variants.
Okay,
so presently, sir, after the third wave, we can able to survive, sir.
And there is one more query. I think you have been answered this. do you think with the coming waves of virus effect will get weaker? Is a question for one of the participants, sir.
Yes, sir. Actually, the research is not yet completed. Like they didn't do fully. Like it can able to treat because the research they have to do nearly 100 to 1,000 patients, isn't it? So only few patients are infected with these kind of variants, Lota, Eta, etc. But presently we can find at least few Delta variants in Kerala and northern parts. So up to Delta, I think we can come into control. It will be declined within few days.
Thank you very much, Dr. Praveen Kumar, for being a part of this Global Virtual Healthcare Summit, sir. Thanks for your time and knowledge to sharing with us, sir. Thank you very much, sir.
Thank you.
Yeah,
thank you very much, sir, for providing an ample opportunity.
Yeah, I'll request you to stop the video so that I request Dr. Rahul Rathod, our next speaker to our second version of Global Virtual Healthcare Summit, conducted by AKT Health Analytics in partner with TAN. Dr. Rahul Rathod is a cluster medical head at Dr. Reddy's laboratory. Dr. Rahul Rathod is a postgraduate medical professional with MBBS and MD with an executive MBA degree from Wellinger Institute, Mumbai. He has more than 16 years of experience in the pharmaceutical industry, biotech, and clinical practice. His key area of interest are big data analytics, real-world evidence, post-marketing, clinical programs, medical strategic planning, clinical development of medical devices, and clinical publications. He has more than 20 clinical publications in national and international peer-review journals, and he has more than 30 global and national phase 4 observational clinical programs, EMR-based real-world evidence programs. Prior to joining to Dr. Reddy's, he has worked as a senior director for medical affairs at Mitra Biotech, Bangalore. Before that, he was giving an increasing responsibility at Bayer Pharmaceuticals as a head of medical affairs and regional medical lead and global medical affairs lead based out of Beijing, China. He earlier assigned with Zydus and Denmark. Dr. Rahul, I request you to share the screen and you can you can start your presentation. I request audience to, if can post Q&A questions, so at the end of the session can be answered by Dr. Rahul. Thank you, sir. You can share the screen, sir. Yeah, Dr. Rahul. Sir, you can unmute, Dr. Rahul. Dr. Rahul, you can unmute. We are not able to hear you.
Okay, my bad.
Yeah.
So are you able to hear me now? Are you able to see my slides?
Yeah, we can able to see you and hear you, sir.
Yeah, excellent. So once again, I would like to thank you, Vishal, for a kind introduction, and also like to thank AKT Health for providing this platform to share my views and experiences in this domain of post-approval evidence generation strategies. Primarily in this second version of Global Virtual Healthcare Summit, and happy to be part of this this version as well. Before I actually start, let me give just a disclaimer that the opinion expressed in this presentation and all the subsequent slide is solely of myself and not necessarily of my company, and surely you can read it on my slide as well. Yeah. There are two objectives with which I wanted to share my views. Number one is how can we work on evidence generation strategies which will complement the clinical development programs. You know, when I started my career early in 2000, I think that time the post-marketing studies were always a post-marketing or post-approval clinical development programs, which is usually run by the medical affairs department. Yeah. However, in last few years, there is a significant change which has happened from just a sequential way of doing a clinical programs, from clinical development to HEOR to the post-approval studies. Now it is more integrated, means even before the product is marketed, the medical affairs start contributing in a data generation activity, which primarily complement the clinical development program. So I would like to share some of those, some of those views and ideas how this can work. And the second objective is how can as a medical affair professional, we can define the therapy development, complete therapy development strategy around the new product which we are proposing to launch in future, keeping patient outcome at the center. Okay, so that is what these are the two key learning objectives with which I'm going to share my views. So I will take I have only around 12 slides which I will try to cover most of the strategic aspects. So I will not go in a lot of operational thing. Mostly what I will be sharing is more of a strategic viewpoints or experiences which I had, and I also picked up some of those learning elements from some of the public domain literature. Yeah. So let me start with very simple. Let me start with very simple slide first. What is a strategy? So simple English definition, when you just do a Google search, you will find out medical strategy. A strategy is primarily a plan of action which is designed to achieve a long-term goal or long-term objective or long-term aim, which we look forward to. And each strategy will have a lot of tactical plans, a lot of milestones, to achieve the aim by which we are doing it. Yeah. Maybe few of you who may not know what medical affair is. So medical affair is one of the important pillar in a pharmaceutical industry, where they are primarily responsible to ensure the medical marketing plans of a new product which is launched in the market is there, so that the product is picked up very well. It's been shaped up into the treatment strategy of management of a particular disease for which that product is made available. Again, the medical affair, if you go back to a few decades, medical affair was considered as more of a support function. Means when I say support function, is primarily to help marketeers to understand, what is the science behind this product, or help the sales team to train them how to sell this product, or to provide them support for the evidence generation activity. From that phase, now medical affair is completely transitioning into a new strategic role within the organization. Why? Because with our medical credentials, scientific know-how, and credibility among the stakeholders. So when I say stakeholders, these are primarily the KOLs, the key opinion leaders, the top level doctors within the country or outside the country. We are emerging as a natural owner of scientific knowledge and the data within the organization and across the entire life cycle of the product. And because of this, it offers us a unique opportunity to principally move away from a support function and force a new role as a primary strategic pillar of the organization, now which can go hand in hand alongside with R&D and as well as a market access. Now market access is very important function within the commercial team, who start working even before the product is launched. Means even in a development phase, early development phase, market access primarily works to find out what is the place of that product in the treatment algorithm of any particular disease for which that product is getting development. And we all know R&D primarily work in identifying that product and developing it into the level where it can become usable by the human beings. Again, picking up this from McKinsey, this is a vision statement in which McKinsey has shared one article which talks about medical affairs vision by 2025, and it actually says that medical affair can drive improved patient experience and outcomes. That means as this new healthcare landscape is starting to shape up, the different types of evidence generation which is required today, like starting from a regulatory perspective evidence generation to the increase in stakeholder sophistication for evidence generation. When I say stakeholder sophistication means when I go with a new product to a physician, they will expect me to compare my product with some other currently available gold standard or a standard of care, or they will expect me to prove that my product has certain benefits. That benefit can be simplistically from maybe the frequency of dose administration to the providing such significant clinical benefit. Clinical benefit can be in terms of safety or efficacy. Yeah. So there is a sophistication in which the stakeholder is now expecting us to provide the evidence for any new product which we launch in the market. And it's also important how we gather, integrate and interpret this data, because I believe that this is going to define the future going forward for any new product or any refurbished product which you're going to launch in the market. So there are multiple elements around it, like there are multiple integrated evidence generation elements like health economics and outcome research. Again, not very relevant in India, but very critical for some of those countries where the products are reimbursed. And followed by that real world evidence, another significant contributor to the currently the evidence generation strategy. I will just touch base upon in my subsequent slide how they are making sense. And we also look at some of those intervention study like phase 3B, where I may go beyond regulatory required studies and try to develop maybe a comparative studies, or may have some studies which will have more expanded duration studies, like just starting, if I am doing a surrogate marker studies for some of the hard outcome, I may continue doing the phase 4 interventional studies to find out more real life, hard outcome, hard study endpoints like rate of mortality or rate of myocardial infarction or a stroke, if that particular drug is falling in that particular category. Yeah. And I feel like medical affairs team, through their deep understanding of the science, data capabilities, their dialogues with the external stakeholders, which involve not only KOL but also the routine also the doctors at a ground level who actually doing primarily practicing or seeing their patient on day-to-day basis. They are very well positioned to understand how to seek evidence required to support the entire life cycle of the product, which will offer an optimized patient outcome. I think that's where medical affair is going to play a significant role in coming future. I believe this point. And again, touching upon this point, I think medical affairs now can get involved in evidence generation early in the life cycle of the product, as you can see on my slide. And just restating what when I started my talk, I think, see, it was a common earlier years that for pharmaceutical companies to consider evidence generation as a responsibilities of individual departments like clinical development department or medical affairs department or market access department doing HEOR studies. And most of the time, these evidence generation were done in a sequential fashion. That means one after another. For example, you first do clinical development studies done by the clinical development, which are primarily for regulatory purposes. Then HEOR primarily for purposes of showing that there is a cost effectiveness benefit of the newer therapy versus the current standard of care. And then the medical affair coming into the picture to do post marketing clinical programs. This is more like a risk averse strategy which we used to follow in the earlier days. However, we have realized, and I will share some of the experiences from some of my earlier companies which are there in a public domain phase, how this has completely changed and how this has completely changed over last few years. Yeah. now it is integrated approach. What do we mean by integrated evidence generation strategy? Means you have a clinical development department which primarily focuses on the regulatory approval piece. Then you have HEOR or market access team who actually parallelly work with finding out what is the market need for that particular product, and parallelly the medical affair who understand the various stakeholders requirement, whether the patient requirement or the KOL requirement or the disease therapy requirement, and accordingly parallelly they work on a strategy which actually talk to each other. And when the product is marketed, you have the complete evidence piece around this molecule which will help me as a doctor to or a patient to pick up that product because it fits in for my treatment. Yeah. And this integrated approach planning involves maybe the randomized studies, real world evidence approaches from all departments, which offers an innovative operating model, and they can go from anywhere from phase 3 study to phase 3B, phase 4 studies. You have RWV, you have patient reported outcome studies, you have disease registries. There are so many different kinds of studies you can actually think about. And why this is important? Primarily because, see, traditionally when this clinical development study happens, it evaluate the new medicine with a sole purpose to get a regulatory approval. And it does not or may not have the evidence that patient, clinicians, or decision makers like payers requires to make the decision to use that product for the particular disease. That's where this entire integrated medical evidence strategy is coming into the existence now. So what are those most common purposes for which we do these studies? So two element to it. Number one is more specific to the disease area. Second is very more specific to the product itself. So when I say what is that is specific to disease area, is primarily, is there any unmet need in a current disease management with the current treatment? Is there any unmet need? So we may do some of those research around this even before the product is getting launched, maybe in an early phase 2 study or phase 3 study. That is the time I actually start working on it to identify, is there any to understand the natural history of disease, what is happening, what is the burden of illness, what is the incidence prevalence, is there any change happening over a period of time, is there any comorbidities which may having some impact, which may affecting the way I am managing that particular which is affecting the way I'm managing the patient with the existing treatment. Does my product which I'm going to launch in future, does it have any beneficial effect on that? To understand the current clinical practice, again, this is very, very important. How currently the patients are managed, what are their treatment gaps, is there any unmet need in that pattern of treatment? Just to give an example, like when this entire Rivaroxaban, which is launched by the Bayer, I think they did an amazing registry. We started even before the product was launched in 2009. So this was like Garfield Day of registry. You can actually look into the website and find out about more detail about it. It's a very long-standing registry program with an amazing way in which this entire registry program was planned to really understand how the patient with atrial fibrillation as treated with currently available anticoagulant therapy, which was primarily warfarin, which was the most commonly used as a oral anticoagulant therapy that time, and what are the treatment gaps, what are the patient level problems, what are the physician level problems. We are able to understand that. And we know, I think these novel oral anticoagulants like Rivaroxaban or dabigatran and all those therapies, how they are able to fulfill that treatment gaps with anywhere between just doing a regular monitoring. Every month you have to do a blood testing, which is not required to be done for this. You have to monitor the food when you are using warfarin, because warfarin has a very high level of food to drug interaction versus the novel oral anticoagulant. So there are a lot of ways in which those treatment gaps were identified, and these were able to fulfill with the new treatment. Again, also you can also use these programs after launch also to understand how the new treatment is getting utilized, and what are is there any outcome predictors. The outcome predictors can be anything from maybe the young age, or a patient with no comorbidity, or patient with blood, if it is an antihypertensive, maybe in certain range of blood pressure, they may be, is there any treatment outcome predictor. This outcome predictor can be positive or negative. What is the population level impact of this new treatment which we are going to introduce? So this can happen before as well as during and after the product is launched as well. We can also look at, and most of the time the data sources for this is coming from a lot of registry data, a lot of payer data, or you can also plan some of those prospective registry program which actually captures the real world clinical practice for that particular disease area. You may also look at a product specific, and most of time these product specific studies are done after the product is approved. It may not happen before the product is approved, but you can consider those in your planning phase even before the product is launched, by understanding the intricacies of that particular product, understanding how the current standard of care is working, and what are the current treatment gaps. Accordingly, you can also decide, do I need to do head studies with the standard of care? Does it really make sense? I need to plan real world, short term, long term safety effectiveness studies, because in clinical program you have a very restricted population in which you are using it, and I may not get a real feel of the product. How when the product is getting used in the real world, it is completely different than the way it has been studied in the clinical, very restricted or clinical programs. What are the long-term clinical outcome studies, which I need to do, which may be beyond surrogate markers. We may also need to look at outcomes which are related to adherence and persistence to the therapy. Please understand, I think when I do a randomized or a controlled clinical study, the adherence rate is controlled by us, by the clinical trial department, by ensuring regular follow-up with the patient. They're coming to us, we checking whether they're taking the medicine on a regular basis or not, because this also impact the outcome of the program. But that doesn't happen in a real world scenario where physician is prescribing the medicine to the patient will take at his own wheels and fancy, so based on how good or bad he is feeling and other parameters. So it's important to see the adherence versus outcomes. Yeah, we may also get to check some of the subpopulation which are not studied in the real world study. We can also look at some comparative effectiveness in different platform, or sometimes we also do patient reported outcomes. Again, patient reported outcomes become a fad in early 2010 and early 2000 and late 2000, early 2010. Yeah, so we need to see, does it fit all, and maybe another area which we can operate in a post approval phase is additional any anticipated new indications, which may pop up during the research. And there are so many examples I can quote where the new indications. I will give an example of crizotinib, which was a drug tested for lung cancer patient, and it miserably failed in the phase 3 studies. So, however, the scientists went in depth and understood, is there any subgroup where this product is working, just to find out there is a 5% of the lung cancer patient has a very specific genetic, these very specific gene abnormalities in their lung cancer, that is ALK abnormality. Okay, and what we have observed that crizotinib works very specifically in the lung cancer patient with ALK, which is around 5% of the total lung non-small cell lung cancer population. Now, crizotinib is a standard of care for that particular subgroup of the lung cancer patient. So that is, sometimes it is important also to really see, can we look at some anticipated new indications or additional indication which can support the development of this product further. When we are working on a strategy, let me tell you, there are so many different kinds of strategy design. I try to enumerate few in my couple of slides before. You can look at, interventional clinical program or an observational clinical program. But just remember one thing, that one size does not fit all. Just to give an example, patient reported outcome research. This is, as I said, this is a very hot topic in last few years. Patient reported outcome studies in psoriasis, which is a skin disease, if you know all well, where you actually see there is a patch, dirty looking patches on the skin, because of some abnormalities in some of the skin layer. Yeah. And when I see it as an open eye, it looks so embarrassing. Yeah. It has so much social impact versus a patient-reported outcome studies done in hypertension. Now, in hypertension, patient doesn't have any symptom, doesn't have any issue. If I'm planning to do patient-reported outcome, it's ideal to do it in psoriasis because there is a patient factor which is significantly contributing to the significantly important, because there are signs and symptoms which he or she can see on his body. He has a lot of social embarrassment. And if I am giving a treatment which actually helps him to clear his skin, his or her skin, it is going to have an important element. And that's why patient reported outcome studies are more important when you have a diseases which is affecting a person in his day-to-day social well-being and other factors, versus in hypertension where patient doesn't see hypertension. He or she doesn't have any signs and symptoms of hypertension that often. And that's where the patient reported outcome may not work there well. So you need to take a call, what type of studies I need to plan during an post-approval phase. I'll just run through a few slides because I think I'm running away of my time. So I think Garfield registry, I also told you some of the registry programs you can do, but again, registry programs are more relevant for a chronic diseases, may not be relevant for acute diseases, unless you really want to figure out what is the incidence prevalences of those particular type of disease. How to work on the strategies. Primarily insights come from multiple sources. I just highlighted few which I have used. Primarily through some of those RWE data sources, you actually can see what type of data gaps are available. Maybe with deep understanding of learning and interacting with the stakeholders, primarily the physicians, where we do a lot of talking, lot of experts, we do a lot of talkings, and we can actually understand what type of data gaps are exist in that particular treatment or a therapy area. And many times even the secondary data sources like published literature, conference talks, where you go and talk and see the interaction happening. Sometimes on social media, patient blog, physician blog, where they post all those challenges or issues they're facing with current treatment or a treatment which has been offered to them. When I'm planning strategically, I have to consider all different types of factors. So the factor is time, by what time I need the data to be available, what resources, funds available with me. Do I need to do investigator sponsor study or company sponsor study? Again, this is very important strategic consideration. So these are individual, company specific, requirement that there are certain kind of study will be done only by the company because they're time bound important for me. There are certain type of study will be done by the investigator, maybe categorized into investigator sponsor strategy. And if some investigator approaches us, and if that their design falls into the strategic planning which I have done, then I go and support that particular study, because investigator sponsor studies are always more economical and more credible compared to the company sponsor study. Yeah. So this last two slides, just remember whatever we are doing, there are only two aim or two objectives. Just keep in mind. Always consider that we are doing this to improve the patient outcome. And always remember that when we are doing these studies, we have to answer a specific clinical question from that particular research activities. And my key messages, I'm just reading out this slide for you all. See, post-approval evidence generation strategies should complement clinical development studies. Again, I would rather remove that word now, post-approval. Maybe we can have an integrated evidence generation strategies where we actually can start even before the product is launched. The purpose should be clearly defined. That is, what is a clinical question. We should identify the appropriate study methodology, considering the resources available to us. And again, remember one thing, that one size doesn't fit all. If something is successful for one, that doesn't mean it will be successful for the other type of disease. And ultimate aim is to improve the patient outcome. Yeah. Thank you very much, and I stop my presentation.
Yeah, thanks.
Yeah.
Thank you, Dr. Rahul, for sharing a wonderful knowledge. And we had one query from our clients. How can you, general practitioner, be a part of integrated evidence strategy to understand and deal the benefits of this new drug?
Yeah. So I think this is very, very important question, which which has been asked. I tried in my last session last year, I think I tried to share how a physician, a practicing physician, can contribute to the evidence generation. See, the importance of real world evidence is improving significantly nowadays, because even FDAs from like say US FDA or EMA and other agencies have started listening to us, and they've started listening to the real outcome of the real world evidence data, including how it can significantly reduce the cost and the time of new product development. So as a general practitioner, how can I contribute? There are multiple ways. I think first thing which I would say is maybe join any EMR, whenever you're practicing, you're doing a practice, keep the good record, electronic records of your patient's medical entire journey, starting from the clinical history, their lab details if they are available, the treatment history, and regularly when they follow up with you, try to keep on updating on it, because this data is going to build up. In India at present, we do not have those integrated systems like the payer systems and all, which is important. But my single statement answer is do EMR, electronic medical record keeping, for your own patients.
Thank you, Dr. Rahul Rathod, to be a part of our session. Thanks for your time and knowledge sharing to us. Thank you very much.
Thank you, Vishal, and I hope there were some learnings for all of you, and it was a pleasure to be here.
Yeah, thanks. We welcome now move on to our next speaker, Dr. Nate Newforth. Welcome, Dr. Keith, to our second version of Global Virtual Healthcare Summit conducted by AKT Health Analytics in partnering with them. And Dr. Keith is a Senior Director for Satara Software, which includes Phoenix, NLME, Piranha, and Trial Simulator. Keith has more than 20 years of global drug development experience and held a variety of positions within clinical pharmacology at Roche, from direct project support role to supporting successfully for NDAs, to a member of global clinical pharmacology leadership team as a system experts of the department. In addition to the expertise in quantitative and system aspects of the drug development, Keith has a passion for understanding and optimizing the human aspects of successful system implementation. Keith has a Bachelor of Science in Pharmacy, the University of Connecticut, and Pharmacometric from Massachusetts College of Pharmacy. Dr. Keith, you can share your screen and you can start the presentation. And I request participants, you can put your Q&A so that the end of the session can be answered.
Good morning. Can everybody hear me okay?
Yes, sir. We can hear you on. You can see the screen.
And the slides are displayed. Excellent, excellent. Well, thank you very much. It's a pleasure to join you this morning to talk about Rsnl. I think over the next 20 minutes, what I'm going to do is give an introduction to this tool. I'm going to dig into a little bit into the actual workflow because I think it's important with respect to how we've designed these tools. Talk about the pharmacometric workflow, talk about some of the challenges of implementing R individually and as an organization, and then give a brief demo of how we've designed these tools to meet some of the requirements of the pharmacometric workflow. All right, so what is Rsnl? Rsnl is a collection of open source R packages. They support pharmacometric modeling work using Certara's NLME engine and pharmacometrics modeling language, or PML, directly from the R command line. We've designed the tools with R Shiny apps that support key workflow steps, and in doing so, the tools themselves can teach the syntax of the R code. And overall, the system has been designed to provide a Certara supported environment for pharmacometric work in R. one of the things I'm going to touch on is why we believe it's important to support this type of work in R. Again, because it leads to how we've designed these tools, kind of the strategy behind it. A fundamental theme is that the tools themselves generate code for you as either a final product or a starting point for something custom. So there's complete flexibility with this approach and a concept of tools that teach. So taking a look at the pharmacometric workflow, the job, what is the job of a pharmacometrics analysis? And, many times it's discussed and kind of the focus is on the execution. However, execution of the modeling work is really only it's a significant part, but it's only one part of the overall job. And you can split the overall job into four main areas. There's a setup phase. This is where a data set is assembled from source data, which may be coming from different areas. This is compiled into an analysis data set. The data set is then explored and QC'd. Errors are corrected, and the analyst usually does graphical analysis to determine starting points for what type of model to begin with, and what kind of approach to take the overall analysis. The next phase is execution. This is where the bulk of the analysis is conducted. It's a very iterative step. Actually, the whole workflow is very iterative. It's a nature of the work. But here is where model is executed. A program puts out a lot of output, tables, listings, and then this output is post-processed. Graphics are prepared to evaluate the performance of the model, and then decisions are made around next steps. How should I adapt this model and run the next model? At the end of this process, the analysis is essentially considered finalized, or a final model is available. Then comes the reporting phase, which is where final outputs for the model are prepared. And deliverables usually include a report plus some data deliverables, perhaps some submission packages for a regulatory authority. And finally, the report phase is usually interchanged with something being done with the model, right? This is why we develop models in the first place. The models are used to solve a problem or aid a decision in the clinical development process, whether it be a clinical trial simulation, to design a better next trial, or to answer a question for an existing problem. So that's the overall workflow. Historically, users have reached for multiple tools to work their way through this workflow. Pick the best tool for the job is generally the approach. One might use one tool for data set assembly, another for data exploration. One might do the model execution and development in one software, but when it comes time to simulate, they move to another software. And this is a very efficient approach for a specific task. There's familiarity with the tool sets, task-specific efficiency. However, this approach requires frequent tool changes, and tool changes can cause significant rework and QC overhead. An example of that would be if you've developed a model in one piece of software and then you switch to another for simulation, the model needs to be recoded. Recoding the model itself is not that difficult. What's difficult and can take time is ensuring that model is actually performing exactly the same way as it did when you developed it in the two separate softwares. So while efficient for a specific task, the cross-task efficiency can suffer with this approach. One of the most difficult parts of using many separate tool sets across the workflow is that iterations can be extremely time consuming. And again, the nature of this work, exploratory development, it's an iterative process. You can expect to need to iterate. And a very simple example of that is an analyst gets to the point where they've drafted a version of a report. They painstakingly incorporated figures into a Word document that they've developed in another graphics package, and they send that report out for review, and they get comments back from the clinical team that perhaps, okay, we don't want to use the drug code for the legends. We want to use our newly determined market name for the report. And, you know, you can be talking hours to days to go back and regenerate in a separate tool and incorporate that. So again, a very simple example, but a very practical example of, you know, some of the deficiencies of stacking together individual tool sets for the task. So why do people reach for tools like R? Number one is a tool like R, which is a programming language, gives you a common thread across the workflow. And what that leads to is exceptional iteration efficiency. If you have your full analysis in one language in one script, you can easily go back to earlier steps, make adjustments, and rerun the code. And full analysis reproducibility is also easily achievable. Using graphical user interface tools, separate tools, it's always hard for users to go back and remember, because they're, you know, they're point and click menu selection. How did I exactly do that at this point in the analysis? With a command line language, it's all documented in a script and very easy to reproduce. So the issue, though, is R is a very difficult language to master, particularly for users that have not previously used a programming language. So despite the difficulty of mastering language like R, our use in pharmacometrics continues to grow. At professional meetings, it you know, commonly comes up. The 2019 ACCP annual meeting, many of the sessions were related to open source software in development, including R. This is from a presentation by Mark Gastonguay, kind of making the point that over time, there's a funnel-shaped expansion of open source solutions being deployed in pharmacometrics. Many of these are R. There is, you know, gross ways to look at this. A Google Scholar search of R and pharmacometrics, looking at hits by year from 2010, shows a addition of 300 publications per year, roughly, since 2010. So a very gross way to look at it. However, the slope is going in the direction that one would expect. Professional organizations are popping up. R and Pharma, probably the most notable. This is a dedicated conference to the use of R and Pharma. It was formed in 2017, and the participant growth in that has been exponential. Looking more directly at R in pharmacometrics, we can look at CRAN package downloads. I think most are probably aware, but R is a software where users can contribute what they call packages, and these are domain specific sets of code that help users in a particular discipline do something, you know, more efficiently. So it's like a dialect within R. It's like a language, an R language within R that users can basically use to do something that they need to do for their domain. And if we just pick a sample of 6, you know, these are 6 popular open source packages. Expose is a post-processing tool that was developed for non-mem. MRG Solve is a simulation tool from Metrum Research. We have an open source nonlinear mixed effects modeling tool, some tools for you know, non-compartmental analysis. TidyVPC is a tool that we actually released to open source, my team, earlier in the year, and it's for a GUI environment for preparing visual predictive check diagnostics, which is a simulation diagnostic commonly used for pharmacometric models. And if we look at, these are daily downloads. So the number of downloads every day over the last 4 years, again, the slope is in the right direction. We see some variability. However, in a discipline where it's estimated there's 1,500, you know, roughly based on professional organization membership, pharmacometricians globally, these are significant numbers, and certainly the cumulative downloads of these, again, specific pharmacometric specific R packages, the slopes are all going, you know, all rising quickly. There's probably other packages we could look at, like ggplot2, some of the differential solver packages, but those would not be pharmacometric specific users only. So I didn't include those in this slot. So what are some of the challenges with the adoption of R? They can be both individual and organizational. From an individual perspective, I touched on this before. Mastery of R requires a programming mindset. In my experience, there are some users who simply don't have this. Extremely talented, developers, clinical pharmacologists I work for, some of them just aren't programmers. However, with the right training, many individuals could become extremely proficient R users, but it takes some work. One of the, another challenge with R is there's many ways as a language, there's many ways to do the same thing. There's over 18,000 R packages on CRAN. So a new user trying to figure out how to use R for pharmacometric work, a big question is, which one of these should I invest in learning? And domain specific use cases are probably the best tool for learning. But where to find these? Fortunately, if you work at a big organization, there's probably colleagues you can speak with. There's always internet searches, but the best example is to be able to find an example piece of code to say, okay, this is what I want to do, and then start from that and modify it. From the organizational perspective, there's a desire for harmonized workflows in processes in general. And the reason for this is to simplify staff training and onboarding, to ensure consistency and deliverables to other business functions. This can be a challenge with command line tools. If you have 10 separate users, using an open source tool like R and delivering 10, types of formatted Word documents to the regulatory documentation group, that group is going to be required to do a lot of additional work compared to if they receive the same, format each time. In addition, organizations, they need domain-specific maintenance and support. Very often with the implementation of R solutions in pharma, you have business users that are assuming key roles in maintenance and support. And this certainly is not ideal. These people are scientists first, not support folks. And it's not an ideal situation, but in fact, in a way, it's driven by the complexity of the domain. While at Roche, I was it's interesting, kind of a funny story. While at Roche, I was a user of S+, and I kind of served as the S+ troubleshooter in my clinical pharmacology department. And one day someone came into my office and said, Keith, I'm having a problem. I looked at the problem, I couldn't solve it. And I said, you have to call IT support. So they left my office. They said, okay, thanks very much. And 10 minutes later, my phone rang and it was IT support. They said, yes, Keith, someone has a problem with their S+. So it went in a big circle. So finding support for these solutions can be an important, issue for organizations, wanting to have sustainable, stable working environments. So the strategy behind Rsnl really is to provide a solution that meets both individual and organizational needs. NLME is a capable analysis engine. PML is a capable modeling language. There are many types of models supported. I provide a link to PML School, which gives a good overview of the types of things that you can do with PML. PML is a shared language between Certara tools. Users can grow into Rsnl from other Certara tools. I mentioned we support key workflow steps with Shiny apps that teach R, and we've designed it to work hand in hand with other R solutions. We have plans to support, you provide expanded workflow support beyond the analysis. So really the work we're doing for report generation is a very good example of that. And in the end, the use of these tools will provide major efficiency gains for the full workflow. From an organizational perspective, Rsnl really adds to the scalable solutions offered by Certara. One can use Phoenix for non-compartmental analysis, which uses the same PML language So a suite of tools that can support both junior and senior users in the department is of importance. You can combine it with our integral solution, which is a cloud-based data repository with 21 CFR 11 compliance capability. We have a tool called Piranha that I'm not going to discuss, but this is a workbench for management of pharmacometric analysis workflow. And implementation of that at organizations leads to easier workflow harmonization. Stability and quality of the packages, really we're building these like a software development company would build them, following guidelines to ensure quality of our packages. And finally, Certara is able to support these packages to provide support for the use of R in the pharmacometric workflow. And that's important. I mentioned my story at Roche. I wish I could have sent my user to a company that could have helped them better. So I think I have. Am I out of time? Do I have 1 minute for a demo?
You can continue, sir. You can.
Okay, excellent. So what I'm going to demonstrate very quickly, obviously in the time allotted, I can't cover all of the syntax in the tool, but I have our command line code, and I'm going to just show you quickly the concept of the tools that teach the Shiny apps. So here we're working at the R command line. We import a data set. One might do some exploratory data analysis. Here we're using ggplot. Again, grammar of graphics is a very popular package in data science. We get a plot. This looks like 2-compartment drug, perhaps. One could expand into other R packages to do further exploratory data analysis. There's a ggquick EDA package. This is an exploratory data analysis package that was developed by one of our consultants in Certara. So we have a long history of actually contributing to open source on our consulting side. That's Samir Muksasi, very talented individual. But for the interest of time, the next step would be to define your PK model. We use, again, this is R speaks NLME syntax. We have a PK model command. Here we define a 2-compartment model, and it takes the default for the absorption phase, which in this case is IV is the default, maps the model variables to the data set, and assigns a model name, which will be a directory that contains the model output. So we run that. We then adjust the model, removing random effect terms from 2 of the parameters, CL2 and V2, adding some fixed effects. These are the general steps of setting up a model prior to execution, and we're going to look at some covariates. So we run this and we print the model, and you get a nice summary of the model. And for those of you who know PML, this is exactly the same modeling syntax that is used in Phoenix Wind Non-Lin. Now, this looks all very complicated, right? So what happens if you don't know the code? This is where the UIs come in. And the first UI that I will briefly show, and perhaps that's all we'll have time for, is the model builder UI. This, again, is an R Shiny app. Everything, the foundation is R. And that's designed, again, so you have the complete flexibility to customize work to your exact problem. And that is the nature of pharmacometric work. What you do next, if you ask any scientist, if you ask them, what are you going to do? How are you going to do this piece of work? And they say, well, it depends. And you ask them, well, it depends on what? Well, it depends. They need to look at the data and interactively decide what to do next. And that's why command line languages are so, so popular. But this interface, just very quickly, there's drop downs. You can very quickly specify the type of model that you are going to fit. Easily over 1,000 combinations are supported. So if you wanted to specify a model, you see the code is immediately updated in this panel here as you make selections. So if I wanted a PK indirect response model with 3 compartments, Michaelis-Menten, elimination, and I want actually a inverse inhibition, you get the idea. The code is immediately updated, and the user does not need to know how to write it. one would step through the next tab, and the tool kind of steps you through building that model. So you'd specify covariate influences. Again, I won't do it in the interest of time. Fixed effects, random effects, block statements, that type of thing. And in the end, of course, you can toggle back and forth to your data set. In the end, one can generate the RSNLME code that produces that model. So it's a way that the tool can actually teach user how to write this code. In the interest of time, I think I need to stop there. I hope that gave you a good example of how we've designed these tools, why we think it's important to support R in this domain. And at that point, I'll stop and take any questions.
Yeah. Thanks, Dr. Keith, for a wonderful session for NLME. I request to post Q&A for the participant. Sir, we have one question for: Is Rsnl are more complex to use when compared to impurity LME?
It's an excellent question. By TNLME, I'm assuming that might be a typo when you
I think so, sir.
You
NLME.
You mean Phoenix
Yeah.
NLME.
Yeah.
Yeah, so Phoenix NLME is a graphical user interface tool. The model syntax between the two tools is identical. Where R becomes perhaps a bit more complex is instead of being able to use GUI type tools to work with the models, you're working with a set of R functions. So again, it is slightly more complex. However, what the gain is the efficiencies across the workflow and some of the types of things that I described, as well as the access to the larger open source community and the really bewildering set of tools that are available to assist various elements of the work. And again, that was one of the main reasons for extending the PML language into R. So it is a bit more complex, yes, but the payoff is worth it, in my opinion. Yeah.
Okay. Thanks, Dr. K. I think one more question is there, sir. Are you having any session coming up with Rsnl in India to help us understand its difference and compare it to NLME?
I'm not aware that we have any directly planned, but this is something that certainly could be arranged. I'd be happy to participate in something like that. Yeah, 20 minutes is kind of a short time to dig into a command line language, but yeah.
Okay. Thanks, Dr. Keith, for participating with the Global Virtual Healthcare Summit and sharing your time and knowledge. Thanks very much.
Thank you very much. Have a good day.
Yeah, same to you. I request Dr. Keith. We welcome our next speaker, Mr. David A. Hall. Mr. David, welcome to second version of Global Virtual Healthcare Summit.
Thank you.
Coordinated by AKT Health Analytics with TAMS. Mr. David is a former medical center administrator with 5 years of agriculture level work, including Masters in Healthcare Administration, MHA at Indiana University in Asian Studies, MA and Harvard University, as well as a Washington DC Harvard Fellowship in the Office of Honorable Lee H. Hamilton. David performed several years of additional studies in management, information systems, and information technology, MIS, IT programming in project management professionals, PMP, with dozens of technical certificates. David has held Fortune 100 and 500 management position, adjunct faculty position at universities in China, co-founder and helped build at least 10 startups, including the first fintech, first AI, and first e-commerce companies. Have coached and mentored dozens of other startups and has founded Time to Travel 45 countries. David is currently the co-founder and CEO of TAM Inc., where he is leading a team to build a global community of doctor and super app platform ecosystem for wellness. So you can share the screen and you can start the presentation, sir. And
thank you for that. Thank you for that kind introduction.
Thanks, sir.
And a good segue from Dr. Nifor's presentation, talking about making software systems easier to use for non-doctors, for non-scientists. Also, we've heard today from many medical doctors, doctor innovators. And what I'm going to talk about is kind of the glue that holds all of this together as part of this digital health movement or digital health revolution or transformation. And that is the doctors, the medical doctors at the center of this transformation, especially leading and innovating in this new norm. So today I'd like to talk about some of the pains that we're experiencing in healthcare. I'd like to talk a little bit about our working hypothesis and then share some of the benefits we believe that will derive from that working hypothesis, and then finish with a call to action, because we cannot do it alone. Everyone here together, especially GVHS, launching this summit. We hope this is a launching pad to this furthering this digital transformation. I'll begin with the pains. Innovating, as we all know, innovating during this new norm is a challenge, to say the least. And not only that, we need to prepare for the next inevitable crisis. We have digital acceleration and converging technologies. It seems everything has gone, as we say, virtual overnight. Acceleration has accelerated, some say, by as much as 5 to 10 years. Virtual care coordination has become a pain. Developing hospitals without walls, and it has exposed the doctor limited and non-involvement in the digital health innovation, especially human-centered design. And we believe that is the key to these pains, and I'll talk a little bit about our working hypothesis in a minute, but placing the doctors at the center, adequately defining digital literacy, because this is all new. What core competencies are required? Nearly every government, NGO, academic institution, and expert has said that digital upskilling for doctors and healthcare professionals is the most important. Yet, there are only a few programs globally and a few approaches to tackle this pain. And I'll talk a little bit about that. And then we have the continuous regulation and push for value-based care, whatever you want to call it. It is improving outcomes, decreasing costs. A lot of doctors see red when they hear value, right? Because they're thinking this means more bureaucracy, more paperwork, and less face time. And so that has become a huge pain. The increasingly complex patient populations with ever increasing demands now for personalization, customization, the experience economy, has even hit healthcare. Patient satisfaction online, doing surveys, rating doctors in hospitals. And this has caused a lot of doctor stress and burnout, especially because we believe that doctors have not been the center of digital health innovation. And this has contributed to a serious pain. And we like to say pain because it is an emotion. We use design thinking, use the term pain versus a problem or an issue. As we know, clinics are down by some tens or hundreds of percent. Some have closed. Payments are being reduced. We had in the US around 47 bankruptcies last year. And of course, it has been a pain throughout the world. And Dr. Neforth talked a little bit about mindsets, the importance of the mindsets. And we believe that doctors as the center need to develop new mindsets, such as design thinking that I mentioned, exponential thinking, the 6 Ds of exponential thinking, and develop digital literacy and the core competencies. Every industry is attempting to do this. Healthcare is very unique. Doctors, of course, very unique, and other non-doctor healthcare professionals. We have about 8 to 10 million doctors in the world. Again, it's been identified as the #1 area of focus. So we need to determine what skills, what digital skills do doctors need to have. So it's very, very different when you move to a new medium, when you move to a virtual world. For instance, what do doctors need to know about AI? We had a lot of technical discussions today. We talked about coding, programming, making that easier. For example, doctors may want to know, how did machine learning and artificial intelligence arrive at a particular diagnosis or conclusion? They want to know what is the logic and the process that technology went through. And then later, they want to be involved in the co-creation and co-design, because they're at the point of care. They are the best ones to be involved in the innovation. Offering doctor digital health innovation upskilling programs, or DIUP. And this needs to be done on a secure, private, safe digital health innovation platform of some type, because doctors learn better. They feel safer if they can make mistakes in a virtual environment. They can fail in a virtual environment. You cannot do that, of course, in healthcare, where patients' lives are at stake. It's very important. And providing real life experiences. As I mentioned, acceleration is accelerating. The famous futurist Ray Kurzweil has said that he sees a new breakthrough every 3 days. And for someone like that to say it, it is true. He spends his life studying it. So we need real doctors, doctor innovators like we've seen today, sharing their real life experiences as trusted doctor innovator facilitators. And we need to conduct more studies. We need scientific models and approaches in this new medium, in this new world. Soft skills have become the new hard skills. And this has been a kind of a transition from the beginning of social sciences, and then SaaS, software as a service, now into soft skills as a new hard skills, and now into moving from the physical world into the virtual world, where everything is turned upside down. We can possibly use some of the scientific models and approaches that are out there, customize those. However, we need to develop new ways of doing things. Change management is a good one. Accelerate, which is a dual processor model, where you can have business as usual on the left, and you can have innovation on the right. Because we know a lot of hospitals are saying, I can't keep my head above water. How can I innovate now? And how can I prepare for the future. And applying these models and approaches allows healthcare institutions, it allows medical associations to be able to do just that when they believe that it's impossible, but they know they need to do it. Deliberate practice, flipped learning, which is preview, on-site or live interactive like we're doing now, and then review, measurement, follow up on the back end to make sure that the digital health skills are being used in daily practice by doctors. We need to create new measurements. A few that I'll mention are digital scorecards. And this is similar to if you look at even social business, social enterprise. They had to develop new measurements. So there are dozens of different measurements and they're coming up with new ones every day. How can I measure the impact? And healthcare is like that. It's an, it's an ethical, moral, wellness is completely different than something in industry. So how can I measure that? And then now how can I measure doctors' digital literacy? And you can do that with digital scorecards, applying Kirkpatrick's model, which is a well-known model in learning and development, reaction learning, behavior, and results. And now even net promoter scores, maybe developing something new like digital value-based healthcare measurement, where you can tie the measurement. And a lot of companies and a lot of people out there now are attempting to do that, to say, what is value-based care? How can I measure that? What's there's systems out there with checklists and things like that, value committees at hospitals to determine, what is value. And this has been since my days at Indiana University Medical School and Medical Center, in the early 90s, working on my master's in healthcare administration. What is the goal of medicine? We were talking about the future of medicine back then. Watching videos on what we're seeing today that is a reality. And now the focus is different on preventative, precision, customized medicine, wellness, holistic wellness. So it's completely different. And digital health technologies can make value-based healthcare a reality with doctors at the center. Doctors from the point of care that know it best can solve their pains better than anyone else. And the benefits to this are safe, secure environment where doctors can feel comfortable and they can learn, they can apply the digital skills that they've learned immediately from real life experiences of other doctors. There's not time for a several year academic program, and they don't need, as I mentioned, to learn. We do have doctors that program in Python, that program in artificial intelligence, but that is not the main goal. We want to create a sustainable, long-term, win-win, innovative system where we incentivize doctors, we incentivize healthcare institutions, and this is going to reduce a serious pain, the doctor's stress and burnout. Doctors feel they are not involved. They haven't been involved. Even the digital giants have acknowledged, IBM Watson Health and Google Health have acknowledged, we didn't involve the doctors. We didn't apply human-centered design, and that's why they failed. So we want to exceed digital value-based requirements, and it's now a reality with digital health, with digital technologies. We can not only meet the requirements, government regulations, we can far exceed those, and we can shift visits to virtual visits, hospitals without walls by as much as 50%. We can improve preventative, holistic, personalized health and wellness with doctors at the center. So I know I'm running out of time. I'll just finish here with a call to action. This is a global movement. We can't do it alone. GVHS has been so kind to have this wonderful summit to bring together, as I mentioned, diverse groups from around the world, doctors, scientists, technologists. We need further studies to demonstrate the direct positive relationship between doctors and digital health upskilling and innovation, and how that affects improved patient care delivery and outcomes. So a lot of people are attempting to do that with, say, for instance, AI. You know, how does AI, if it reduces risk of nosocomial infections, hospital-acquired infections, adverse drug reactions, things like that with AI, you know, who will pay for that? The payer, the provider, the patient, you know, they're reducing risk. So we want to measure the impact that doctors are having together with the AI. So it's not, you know, doctors are not going to be replaced by AI, but doctors with AI and healthcare institutions with AI will replace those without. So we need further development of doctor digital innovation upskilling programs. There are only maybe 15 or 20 globally, and this is the number one most important area. So we need more of these programs. We need to define the digital literacy, the core competency. We need further studies to show how we're decreasing the doctor's stress and burnout, how they're, you know, volunteering, you know, to be part of these acceleration teams. They're self-actualizing on Maslow's hierarchy, and, you know, it's reducing their stress. They feel involved, they feel in control, and they're making life better for everyone. And we want to improve development and measurement related to digital health innovation. We need to conduct more design thinking interviews, which we've been doing for several years. We need more of them with the key design personas, especially medical association leadership. They've always been the traditional provider of continuing medical education, professional development, and it really is a moral imperative for medical associations and healthcare institutions to digitize, to provide the digital skills and platforms for their doctor members. And healthcare institutions, C-levels, whether they have a value committee or now there are a lot of new titles that have some type of digital or innovation in their title. So that's my call to action. I will end there. If I have a minute for questions, I will open it up to questions. I may have gone way over. Thank you very much.
Thank you very much, Mr. David. We have a few Q&A questions from one of our client attendee.
Yes.
How are you working out with your idea to Indian doctors? As India, it is difficult to collaborate with doctors with artificial intelligence.
Yes. Could you please repeat that? With the working with Indian doctors?
Yeah.
Yes, yes, please.
I'll read out once again. How are you reaching out with your idea to Indian doctors? As in India, it is difficult to collaborate doctors with artificial intelligence.
Yes, absolutely. That's everything we're about. Us particularly, we're a US-based company. We're an extended global team of about 80. And we have locations, innovation centers. We're developing smart clinics in Taiwan, in Japan, and also now in India. And that's one of the reasons I'm part of this summit today. So it's exactly what we're doing. And these programs, virtual programs that I'm suggesting, digital innovation upskilling programs will give doctors in India the digital literacy that they need to be involved in the innovative process. And it will give them the digital literacy and the digital skills that they need. As I mentioned with AI, it will provide them with exactly what they need. You know, what data do I need? And then how do I design the experiment? And then, you know, what's machine learning doing? How does it come up with the diagnosis? So this will be from a doctor perspective, especially. And yeah, India is one market that we are working in, expanding in, and we welcome additional doctors there to work with us from India.
Thanks, David. And we have another question. Have you rolled out any program for healthcare institute to have them understand the importance of healthcare digitalization, and how has the response been?
Yes, it's, you know, everything went virtual this year. So this is very new for everyone. we shifted this past year. We've been working on this for many years with the converging technologies. Digital health innovation has gone virtual overnight. We're getting overwhelming response from large clinic chains. We have found that the medium-sized hospitals are the ones that are taking advantage of these types of programs and this part of this movement. The roughly 100 to 400 or 100 to 500 bed are coming to us and saying, we can't do this alone. This type of out of the box ecosystem will help us to develop, and it's a 3-year. We have a 3-year plan to phase in a sustainable ecosystem for medium-sized hospitals. Clinic chains are coming to us. I'll think of one recently that we're talking to. They came and they said, yeah, we want our doctors involved in the innovative process. We're not quite sure what digital skills they need. We have a lot of digital systems. We know that we want them to upskill. We know that we want them to have innovative and design thinking mindsets because we want everyone on the team to be involved in the innovative process. And so, we really co-organize, we co-work to co-create. And that's really what design thinking is about. We co-create together with them based on their specific pains.
Thanks, Mr. David. I hope because of COVID, definitely it brought digitalization to the real world in a quicker way. Because of COVID, we have been stay to our homes only because of treatment is it's an no man's world. So because of digitalization, definitely it has been provided the solution to most of the people across the world. So most of general cases also get solved because of through telephonic we have been able to connect to our doctors and try to get the medications as per the needs. And I hope can come up with different tools and products with all across the world community getting together with one digital platform and coming up with a proper solution for as per the country specific needs. Thanks for your wonderful session, Mr. David.
Thank you.
Yeah.
Pleasure being here.
Yeah. Yes.
Thank you.
Yeah. I request you can stop the video, Mr. David. Thanks. Thank you very much. And we have our next speaker, Mr. Dr. Chetan. But unfortunately, because of some unavoidable circumstance, he was not able to do this session today. And I definitely thank you, everyone, reaching to an end of this wonderful session of Global Virtual Healthcare Summit 2021. A very warm thanks to our esteemed panelists to give us and the audience their valuable time and knowledge. We consider ourselves lucky to be listening and learning from you. A huge thanks to our attendees as well for being a part of our summit and making it a huge success. We will see you again next year with a bigger summit, hopefully in person. Have a wonderful day ahead. Thank you very much. Thanks, sir.